Hello, dear students, you are studying about excretion and osmoregulation. In this topic, particularly, you are going to study about the urine formation. So, first of all, you will study what is the definition of excretion. See here, excretion means collection, separation and elimination of nitrogenous waste substances such as urea, uric acid, guanine. These nitrogenous waste substances are removed from the body fluid. The process is called as excretion. And what is meant by osmoregulation? Osmoregulation means a process which maintains water and salt balance in the body fluid. बॉडी फ्लुइडमध्ये म्हणजे आपल्या ब्लडमध्ये पाण्याचं प्रमाण आणि सॉल्टचं प्रमाण कॉन्स्टंट ठेवणारी, बॅलन्स ठेवणारी जी प्रोसेस असते तिला आपण ऑस्मोरेग्युलेशन म्हणत असतो. आणि आपल्या बॉडी फ्लुइडमधलं म्हणजेच ब्लडमधलं, लिम्फमधलं जे नायट्रोजनस वेस्ट सबस्टन्सेस आहेत जे रिमूव्ह केले जातात त्या प्रोसेसला आपण एक्सक्रीशन म्हणत असतो. तर तुम्हाला माहीत असेल एक्सक्रीशन किडनीच्या द्वारे केलं जातं. ह्यूमन बीइंगमध्ये एक्सक्रिटरी वेस्ट सबस्टन्सेस ब्लडमधून सेपरेट करण्याचं काम किडनी करत असते. बघा, युरिया इज फॉर्म्ड इन द लिव्हर बट युरिन इज फॉर्म्ड इन द किडनी. सो फर्स्ट ऑफ ऑल यू विल लर्न अबाउट द किडनी. सो in our body this excretory system is present, which consists of left kidney, right kidney, ureter, urinary bladder and urethra. This kidney help in urine formation, not in the urea formation. Most of the students make mistake during the examination that urea is formed in liver, not in the kidney. Urine is formed in the kidney. How the urine is formed, that you are going to learn here in this topic. You know, kidney consists of millions of microscopic tubules. These microscopic tubules are called as uriniferous tubules or nephron. In the NEET exam, the question will be asked, what is the structural and functional unit of kidney? In the kidney, this uriniferous tubule or nephron is the structural and functional unit with the help of which urine is formed. So, this is the structure of this uriniferous tubule. It consists of Malpighian body, PCT, Henle's loop, DCT, collecting tubule, this peritubular network, etc. So, with the help of this uriniferous tubule, how the urine is formed, that you are going to learn. So, here in this uriniferous tubule, there are 3 steps occurs, and these 3 steps are responsible for the urine formation. See how the urine is formed. Urine is formed by 3 steps. Number 1, ultrafiltration. What is ultrafiltration? Filtration of blood under great pressure at microscopic level. Why it is called as ultra? Because this filtration takes place at a electronic microscopic level. So this filtration is known as ultrafiltration. This ultrafiltration takes place here in the glomerulus. Therefore, this filtration is called as glomerular filtration. Now you will study in detail about the ultrafiltration. Where the ultrafiltration takes place? Ultrafiltration occur here in the glomerulus. How it takes place, that you have to study. These afferent arteriole carry more blood in the glomerulus and efferent arteriole carry less blood from the glomerulus. See, keep in mind, this afferent arteriole is larger in diameter and efferent arteriole is smaller in diameter. Therefore, afferent arteriole carry more blood here in the glomerulus and efferent blood vessel drain out less blood from the glomerulus. Therefore, a pressure is created here. This pressure is called as a glomerular hydrostatic pressure. Under this pressure, blood filtrates. See here. Here the blood filtrates under this glomerular filtration pressure. So, this glomerular hydrostatic pressure is about 60 millimeter mercury. This pressure is created due to difference in the diameter of afferent and efferent arteriole. This afferent arteriole is larger in diameter, efferent arteriole is smaller in diameter. Afferent arteriole carry more blood in glomerulus, efferent arteriole drain out the less blood from the glomerulus. Therefore, this pressure is called as a glomerular hydrostatic pressure. This glomerular hydrostatic pressure is about 60 millimeter of mercury. As you know, Newton's law, third law, to each and every reaction there is an equal and opposite reaction. In the same manner, to oppose this glomerular hydrostatic pressure, an opposite pressure is created here. This opposite pressure is known as the BCOP and CHP. BCOP means blood colloidal osmotic pressure and CHP means capsular hydrostatic pressure. Both these pressure opposite the glomerular hydrostatic pressure. So, this blood colloidal osmotic pressure, that is BCOP, is about 30 millimeter of mercury. How this pressure is created? This pressure is created due to presence of albumin in the blood, presence of proteins in the blood creates the blood colloidal osmotic pressure, and which is about 30 millimeter mercury. In addition to that, another opposite pressure is created by the pressure present around the glomerulus, that is called as capsular hydrostatic pressure, which is about 18 millimeter of mercury. So, total opposite pressure formed here is 50 millimeter of mercury. Therefore, net filtration pressure means effective filtration pressure. This effective filtration pressure is equal to glomerular filtration pressure minus opposite pressure, that is 60 millimeter mercury minus 50 millimeter mercury is equal to 10 millimeter of mercury. Under this pressure, blood filters. All the components of blood get filtered from this glomerulus except the blood cells, blood proteins and large proteins from this glomerular filtration slit. Otherwise, each and every component get filtered. Only the blood cells, blood proteins not get filtered because size of the blood cells, size of the blood protein is larger than that of this filtration slit. Therefore, all the components of the blood get filtered here. So, this filtrate is called as a glomerular filtrate. This glomerular filtrate contain useful as well as harmful substances. From this glomerular filtrate, useful substances are reabsorbed from the filtrate into the blood back and the urine is formed. From this glomerular filtrate, when comes here in the PCT, Henle's loop, DCT, the required substances are absorbed back here in the blood vessels and the remaining non-required substances are kept here in the filtrate, and from that urine will be formed, and that process is called as selective reabsorption. So, can you define selective reabsorption? Absorption of useful substances from the filtrate into the blood process is called as selective reabsorption. Selective reabsorption is again of two types: active reabsorption and passive reabsorption. Active reabsorption means the reabsorption against the concentration gradient, and passive reabsorption means the absorption of the substances against the along the concentration gradient. Thus, the substances are reabsorbed from the filtrate into the blood. In addition to that, there is another process which is called as a secretion. Here, due to secretion process, harmful substances, non-required substances which are present here in the uriniferous tubule are secreted back into the filtrate. That process is called as tubular secretion. What is meant by tubular secretion? The substances which are present here in the blood, which are not filtered during the ultrafiltration. The substances which are secreted from the blood, secreted in the filtrate process is called as secretion. This tubular secretion is the additional process, supplementary process of ultrafiltration. The substances which are not filtered during the ultrafiltration are secreted back into the blood during the tubular secretion. And what is selective reabsorption? Reabsorption is the opposite process to that of the ultrafiltration and secretion. Thus, the urine is formed here in the nephron, particularly here in the uriniferous tubule by these three processes. What are their names? Ultrafiltration, selective reabsorption, tubular secretion. These are the three important processes which are required for the process of urine formation. Thus, you have studied the process of urine formation here in this topic.